Hi, everyone. My name is Alhagie Cham. I'll be talking about metagenomic studies of the rose rosette. In this project, we're working to understand the role of host-associated metagenome in rose rosette disease. We outlined some of the progress that we have done for these studies, and then what we're expecting to do, and sort of discuss results from previous studies that are similar to what we are expecting from the future. So we have done field sample collections, also evaluation of disease severity, and then some of sampling processes and storage, and also DNA extractions. So we are expecting to do NGS library preparations, sequencing, and also the multiplexing and quality filtering, also taxonomical profiling. To understand metagenomics for our lab, we have biological questions that drives our research. Basically, we want to know, does the metagenome modulate plant defense response? Can modeling plants microbe interaction improve statistical power? That is for GWAS and predictions. So, and then also can selecting enrich alleles underlying recruitment of beneficial or disease suppression organisms. So for this project, we started working on collecting samples from 12 checks, where we collected six samples for each of the checks across the 6 blocks with an augmented RCBD design. So for the biparental population, we collected a total of 105 samples, and that is the modern fire glow by Miracle on the Houston. So to familiarize ourselves with rose rosette disease, we made a field visit with Mark to look at the field condition and look at the disease severities, and then so from there, so we could make a score of the disease severity. And then to perform the disease severity score, we got a scale of the description that was reported by Young. So that is from 0 to 3 scale. And then we also expecting to get some data from Sarah to incorporate, and then with this scoring that we're doing. All right, so as part of the story, we wish to capture the phyllospora associated metagenome, and that is we do a tissue wash to capture it, and then we do that in a wash solution that we prepare from double distilled water with Clorox and twin 20 at 1%. So we did this by adding 20 milliliter of the wash solution to the tissue in a falcon tube, shake it for 5 minutes with the hand, sort of, and then put it in a shaker for like between 25 to 30 minutes, and then sort of centrifuge it, and then discard the sobrenatant, and then add glycerol to store it for future use. So for the endophytic DNA and RNAs that are enriched in the tissue, in order to capture this, we do what is called the disruption of the tissue. With this, we sort of blend the leaves with the small branches that were collected from the field in order to break the tissue, and then add glycerol to the samples and store them for possible DNA extraction. When we are doing the DNA extraction, we take 1 milliliter of this combination, and then sort of spin it down, and then discard the sobrenatant, and then from there use it for the DNA extraction. So also for the DNA extraction, we use the CTAP SDS based extraction protocol that was developed by Cham, and then we have a modification of this protocol for capturing better genomic DNA. So as far as what we have done in this project and what we wish to do and continue doing, we were waiting for an enzyme that took long to come, so we were stuck in sort of NGS library preparations. So we also have an outline of how long each of these steps is going to take, or how long it will take if you have to use this method. So for the NGS library preparation, it usually take like 2 to 3 days, and for the sequencing and to retrieve the data, take like 10 days. And then for the demultiplexing and quality filtering, takes like a week. For the taxonomic profiling, it takes about a week too. So to work on all this, we have softwares developed in our lab, as well as NGS library sequencing protocol, library preparation protocol developed by my boss, Dr. Olukolu, which is the from where we can see the workflow of this OMSIC library with the softwares for the metagenomic studies that we have that were previously developed in our lab. So the first one is the NGS Composer. That's our automatic pipeline for demultiplexing and quality filtering. And then the other one is the GPS app, which is for variant calling and filtering. And then the next one is the QMedi, which is for taxonomic profiling and microbiome profiling. So in studies that we have done and that we tend to present here to give idea of what we are expecting from the results of the studies that we're doing right now with ROSE, is we've done this metagenomic sequence. Like we believe that metagenomic sequence as a tool for quantifying insects, microbes, mites, and then viruses by quantifying as a proxy for phenotype using molecular phenotyping. So we could use GWAS to identify candidate genes that limit herbivory of insects or viral infections as well. And then we can also use genomic prediction to evaluate the prediction accuracy and quantification and its biological relevance, and also its utility in for breeding. So we also use correlation network analysis to identify biological relevances and then unknown interactions within the microbial host. So this proved to us that with the analysis that strain and species level resolution is crucial for microbial analysis, where we capture more microbial host than genus, family, and resolutions. So in this study, we studied two different populations. That is the association or diversity panel, and the other one is the DC biparental population. For the association diversity panel, on the left is the whole microbial community of the leaf metagenome, where we have species of important interest, as well as insects that are present in this leaf metagenome associated. So when this metagenome is subsetted at 5%, we found an insect of our interest in this study, which is Bemisia tabasi, to have a positive correlation with endosymbiomes of these insects. Also have other positive correlation with other microbes, but not only positive, but also some negative interaction with some microbes in this. So the positive interactions are mostly the strongest, which is the interaction between Bemisia and its endosymbiont. This correlation efficiency were up to 0.8, which is a strong positive correlation between Bemisia and its endosymbionts, which shows the sort of an interaction that exists positively between Bemisia and its endosymbiont. So Similarly, when we look at the DC biparental population, for the whole metagenome, we found much more species, species of different functions in this microbial community. And then when we subsetted the arthropods, we also found more arthropods than the other population. And when we look at the correlations, also we found the same positive correlation between Bemisia and its endosymbionts. So we also found a positive correlation between Franklinella, which is a trip with a trip SP. So as a sort of a relationship between these two. So to look at the candidate genes and the QTL profile from the GWAS, after doing the candidate gene selection, we have a list of candidate genes that are possibly involved in Bemisia herbivory limiting, which we found a candidate gene that is ACC oxidate, which is involved in ethylene signaling and also in limiting insect herbivory. So these genes, after the QTL profile, show that the position and the strong link in this gene in chromosomes positions, so how best they participate in this interaction when it comes to the Bemisia, which is a sweet potato pest that directly interact with this plant. So we also do genomic prediction using metagenome as covariate and for the two populations. And then we realized that when using metagenome as covariate really helps in the prediction for Bemisia, which is a sweet potato pest. But if we, after doing the prediction ability for Oxypus olens, which is a non-sweet potato pest, but and a predator that interacts with insects within this environment, the predictions were not great. So when we use model dosage, we could realize that model dosage also have effect on prediction abilities. So we also look at the prediction for viruses, where we realize for the two populations, when we look at the sweet potato Barnaby virus, between the two populations for the association panel and the DC biparental population, we realized that the association diversity population, the predictions were much better when compared also to previously with Bemisia, also showed that predictions were better for association diversity panel than they are for the DC biparental population. So when we look at the marker density in prediction ability, we found that for the Bemisia in diversity, the predictions were better than for Bemisia in the diversity in the DC biparental population, which coincide with what we previously have when we use metagenome as covariate. For the marker density, we can prove that having higher number of markers will predict better for us. As you can see, mostly the predictions peaks at using 10,000 markers, but also between 4,000 to 10,000 markers, predictions abilities are not very bad. So when we also look at the prediction using number of markers or marker density, we for the sweet potato Barnard virus, we can see that the predictions for diversity are really much better than the prediction for the DC biparental population. As previously explained before, for the Bemisia, we believe that this As we previously reported also that predictions are better in biparental population than diversity, which we believe have something to do with genotyping, which can be a genotyping problem. Well, for the conclusions of this talk, we believe that the host-associated metagenome plays a possible role in host phenotypic expression, and often offer a breeding strategy that accounts for the genomic host metagenome environment. And whereas the modeling of multi-pattern interaction increases statistical power and lower false positive rates in GWAS and genomic predictions. Also, correlation network analysis identify biological relevance on non-interactions, identify potential beneficially and disease suppressing organisms as well. We want to thank everybody, and then thank our collaborators and members of our lab. We want to thank Mark, who just left, and then our collaborators from NCU, some of the former students from our lab in developing some of the software that we are still using as part of our projects that we have in this lab. I thank you very much, and I'll take any questions from here, and then my boss will also have some questions to take. Yeah. What was the genotype that you used and the phenotype that you want to go up to?
So my question for that study that he was showing, the GWAS, was what was the genotypes that he used and what was the phenotypes that he used to do the GWAS?
Okay, I think some of the questions my boss will also come in. So can I skip? So yeah, he want to know.
So obviously we didn't collect phenotypic information for Bernicia. So we're using the title level in the metagenome as a proxy for the phenotype. And that's the same thing for the virus. So it will be similar to if you're doing RT-PCR to check for viral titer. It's the same thing. And we do have cases where, for example, we have fusarium disease, root rot in sweet potato. So we're able to do both the phenotype and use the titer levels of Fusarium proxysporum ibatatus and compare both analysis. You do get more QTLs when you do GWAS on the organism itself, the pathogen titer, but we also get overlapping QTLs between the symptom and the pathogen. And for the predictions also, we get similar estimates.
So when you do this on road, you'll be able to pick up viral titer and mic titer or whatever.
Yeah.
You're going to do those QTL.
We're confident we're going to pick. Okay. Let me take.
Well, yeah, like he said, for that week, we are very confident we're gonna pick some of those are very important for identifying the rows, and then also we'll be able to get genes that could be important for limiting mites, sort of mites interaction with the plant.
And we're fairly confident we'll see mites.
Yeah.
The question is going to be the viruses. So what we're actually doing with the prep is we actually make one prep where we treat it with RNAs, and then we make another prep where we don't treat with RNAs. And just from what we've done previously, whenever you don't treat with RNAs, you basically carrying over RNA and then we tend to see viral RNA. But if you treat with RNAs, you basically deplete all the RNA, most of the RNA, and what you really just find is DNA-based viruses, right? So fingers crossed, we're hoping we capture the RNA, because we're going to be interested in RNA virus. So we're hoping that we'll be able to profile that effectively. But we do have success capturing RNA viruses in other libraries we've made. Yeah.
So you found greater accuracies with the specific panel versus the traffic population. Is that a technical outcome or what's going on?
I'll take it. That surprised us.
Yeah.
Okay. So Oscar's question is, is it typical to find better genomic prediction accuracies in a diversity or multi-parental population than a biparental population? Oftentimes people claim that they get better predictions with biparental populations. And if you look at breeding, genomic selection practices in industry, they try to limit the amount of parents they throw into their breeding programs for that purpose. So I was stumped, but I think what we see might be valid. So one, if you think about genomic prediction models, the assumption is that you're modeling small effect QTLs, right? It's not that robust for large effect QTLs. So you'd expect that in a diversity panel where you'd have a lot of QTL, you'd expect that most of those QTL will be small effects, small effects, right? And you can tell from your GWAS that you really pick things up that are more than 5% in your GWAS, right? So just from that perspective, I think it makes sense that you actually have better prediction in a diversity panel. I also think one of the problems why people don't do well with diversity panels is genotyping accuracy. If your genotyping has a lot of error, then you're going to predict badly. And in biparental populations, you can clean up your data better with by testing for segregation distortion. So I think the reason why you see better performance is data quality, I think. Yeah.
Okay.
So if I'm understanding correctly, that you're using things from the metagenomic analysis to get amounts of mice, amounts of virus.
Yes.
Are you also using that same GPS prep to get your dosages?
Yeah.
For your
Yeah, so the metagenomic profiling is quantitative.
Okay.
And in fact, that's what we're trying to show in the predictions. So we basically mask some of the quantifications as if we don't know them. And then basically say, if we use genomic data, can we actually predict those values back again? And we've seen that we're doing well by like 70%. So it gives us confidence that the quantitation in the metagenome profiles is accurate. Any more questions?
All right, thank you.